Okay. Welcome to another event of the Science of the Human Past, the Initiative for the Science of the Human Past at Harvard, in collaboration with the Getty, which has been the underwriter of much of the research that you're going to hear about today, and with the Max Planck Harvard Research Center for the Archaeoscience. Our theme today, Ancient Empires, Ancient DNA, Connecting Classical Civilization with the Near East. A challenge in many, many respects. You're going to be hearing today from these distinguished advanced and early career scholars and scientists who have been working to produce completely novel information, completely new evidence for the ancient world in the forms of the genomes of the ancient people themselves. Peter van Domelen is the Joukowsky Family Professor of Archaeology and a Professor of Anthropology at Brown University. Alison Barton and Victoria Moses are what I think of as the dynamic duo in the Getty postdoctoral fellowship that has been going on here for 2.5 years. And Reed is in the final stages of archaeogenetic, philological, archaeological study of mobility and migration in the late Roman Mediterranean. The Science of the Human Past aims to bring together different ways of approaching human existence, scientific, archaeological, historical, philological, linguistic, etc., and to bring together people who might otherwise never meet and have them talk. We've just had a fantastic afternoon of people who might never otherwise have met brainstorming about these findings. So it's something that is extremely productive as well as a lot of fun. The Science of the Human Past is overseen by a steering committee, which, as you can see, is recruited from across the university in various departments and schools and divisions. And tonight, we want to ask the question about what kind of new light can we expect on ancient empires from ancient DNA. We're going to talk about 3 ancient empires or world systems or entities, worlds, if you will, the Assyrian or Neo-Assyrian, the Punic, and the Roman. We're going to ask whether ancient DNA can give us any new insights, any new clues, any new data on the interaction of the Near East with classical civilization as we know it in Greek and Rome. Can it illuminate the people themselves who were these ancient empires, who lived in these ancient empires and gave them life and death? We have 2 powerhouses who are participating in this grand project, the laboratory of David Reich here at Harvard, and the laboratory of the Department of Archaeogenetics under Johannes Krause at the Max Planck Institute in Leipzig, our partner of the Science of the Human Past within the Max Planck Harvard Research Center for Archaeoscience. And Reed Morgan's work that you'll be hearing about has been done in that laboratory there. David Reich is somewhere in the offing. I don't know if it's possible to splice him in. No, it's not. He was going to make a comment, a word of welcome. The climate conditions have imposed a certain degree of improvisation, but I'm very pleased to say that so far so good with the talented team that we have. So the challenges for all the speakers who are undertaking this are many. First of all, simply from geography. The environmental conditions in the Middle East are not good for ancient DNA. It simply doesn't preserve well there. The geopolitics, if anything, make it even more challenging. And there's little precedent for success in the archaeogenetic literature. These are the inherent difficulties coming from the region and the character of the samples that we're looking at. The disciplinary difficulties are just as big. We have archaeology and we have genetics, 2 very different disciplines with different tools, with different methods and different questions. And so far, there's no one out there who practices them both. But thanks to the generous support from the Getty, this has given a unique aspect to this research project in the form of our dynamic duo of Alison Barton, who was a Getty postdoctoral fellow here in this project with the Science of the Human Past and Max Planck Harvard Research Center, who is in the Department of Human Evolutionary Biology in the David Reich Lab after doing her PhD in genetics in the medical school. Victoria Moses is an accomplished Roman and pre-Roman zooarchaeologist, Iron Age zooarchaeologist, who presently holds, after her time here, a postdoctoral research associate in archaeology at Brown University after having been the archaeology pair of the dynamic duo under the Getty postdoctoral fellowship. This was a completely unique effort to achieve two goals: to expand the archaeological lens by using ancient DNA to explore the interaction of the Near East and classical civilization through the genomes of people who were these two civilizations, who were part of these civilizations, and at the same time to create something perhaps even more lasting, a duo, an archaeogeneticist paired with an archaeologist to lead the research project and to teach each other their respective skills. So we have today, thanks to this 2.5 years of work together, we have two bilingual specialists who are bilingual in genetics and archaeology. One needs no further proof for this, I think, than the fact that Vicki Moses' first course being taught at Brown University is in ancient DNA and the ancient world. So it's been a remarkable success in that respect as well. So geopolitical headwinds emerged almost as soon as we set out on this from terrible conditions in the Middle East. Was it possible to obtain new samples from the region? The answer is basically no. And so we were forced to make virtue of necessity and turn to legacy collection, to samples from older excavations that are sitting in museums that we can get, well, at least we can be in contact with. Could we get access to these samples? If we could get access to them, would we be allowed to experiment on them? If we were allowed to experiment on them, was there any chance that they would yield any usable ancient DNA? These were really big challenges and took a lot of time and a lot of effort, a lot of patience, a lot of diplomatic flair, and a lot of cooperation among us to get there. And tonight, we want to present to you the first fruits of some remarkable successes. We're going to talk about the empires in kind of a chronological order, from Neo-Assyrian to Punic to Roman. And I want to point out that much of the previous work of this archaeogenetic character in the ancient world has looked at big regions like central Italy, like Rome's Danube provinces, and attempted to show the big picture of the genetic population structure of large regions. Tonight, we will have another beautiful example of that in the work that Peter van Dommelen was so deeply involved in the Western Mediterranean, the Punic world or the Punic Empire. But tonight, we're also going to share with you the result of highly focused experiments, focused in time and in space. The fall of Nineveh is where we're going to start. We know about it from the Greek historians, from the Bible, from cuneiform tablets, and from archaeology. For the fall of Nineveh, Vicki Moses is going to take us to one gate and one day in 612 BCE, kind of Neo-Assyrian Pompeii, if you will. And we're going to add ancient DNA to the picture, and you will be interested in the results. Peter van Dommelen will speak next, and he's going to take us into his fantastic paper, led by Harold Ringbauer, in which Peter was one of the leading archaeologists, and he is a great specialist in the Punic world, in the archaeology of the Western Mediterranean, particularly Sardinia, and of the relations between indigenous and colonial worlds in the first millennium BCE. So he's going to talk to us about this important results about the Punic world. Whoops. We're going to move on after Peter's presentation to a short presentation on the latest information on a project going on right now on the Punic Empire, new samples, new work being done on Tunisia, which of course is the heart of the Punic Empire as it was its capital in Carthage. We will have a fourth presentation from wonderful Allison on Roman Libya. And we're going to see what people look like in Roman Libya in two different parts of this important province on the African coast. You can see where it is on our mapping past societies. And finally, we're going to conclude with some final thoughts by our dynamic duo of Allison and Vicky about overall results from the project so far, about the use of this novel tool, identity by descent, IBD. You'll be hearing more about it, but it's essentially parts of the genome that are the same, identical in individuals, and in turning all this data into something that the average archaeologist and historian can do something with, and the general public, that is mapping Roman genomes in mapping past societies. So, with no further ado, I would like to turn the floor over to Vicky Moses, Dr. Vicky Moses.
Today I'm going to be sharing some results from the beginning of our chronology of this big project where we have multiple different sites, and at the end of the day all of these sites relate back to these ideas of we have changing empires that we know so much about. It's built a sort of imperial culture that spans broad swaths of land in the Mediterranean and the Near East. How does this look like for the everyday person, the populations, and can we see that through ancestry or other genetic markers? And so in this case, as Professor McCormick said, I'm going to be zooming into one moment in time to a small group of individuals from a very unique archaeological context. And before I begin on this presentation about the paleogenomic evidence from the fall of Nineveh at Halsey Gate, I want to thank my co-authors who embarked on this project before I even joined these teams, Michelle Shamounpour and Aryeh Schaus. They are absolute experts in this topic, both from the genetic and the archaeological standpoint. And so it's been a real pleasure to learn from them and to contribute my focus more on the archaeological context itself. And then they are very innovative in this way that they're thinking about these new data that are just completely unprecedented in this region and time period. So we are looking at Nineveh, the final capital of the Neo-Assyrian Empire, and we're looking at archaeological context from the Halsey Gate at the moment when Nineveh was falling. So this is right at the end of a multi-month siege by the Medes and Babylonians and potentially some other enemies of Nineveh. And we are fortunate that, well, unfortunately for these individuals, it's a harsh moment in history, but in 1989 to 1990, David Stronach and his team excavated the remains of multiple individuals, 12 individuals plus a partial human skeleton and a horse skeleton that were part of this destruction layer in the corridor of Halsey Gate. And we're very grateful that to be able to work on this project from the late David Stronach, and Michelle Shamounpour was able to speak with him at length about the interpretation of this context. And based on the skeletal remains, the artifacts, the stratigraphy of this, we are looking at a destruction context with several individuals who were slain in this moment where there's a cobblestone pavement. These individuals with severe skeletal trauma, these individuals were both adults and individuals as young as 10 months old. And they were covered with burning rubble, burning debris, beams, bricks, and many other evidence of destruction of this corridor. And so here we're looking at the excavation area is in red, and I'm about to show you a bird's eye view of what the skeletal evidence looked like there. So here we have the remains, and as you can see, okay, so at the entrance of the corridor, we have part of an individual and part of a horse, and this is where the excavation was limited, and so there's not much data from that part of the area of the exit of the gate. But here we have 12 individuals, and from them we were able to extract DNA from 6 of these individuals, both adult and subadult individuals. And I'm going to walk through some of the skeletal evidence before we go into the genetic findings. There were over 20 artifacts that were associated with weaponry, and these were in the style that was favored by the Medes, so more it aligns with the historical evidence that we have. And all of these arrowheads were pointed into the corridor in an east-west orientation that aligns with them being shot into the corridor by these besiegers. Some of the other evidence iconographically in the ceramics, as well as the C14 date, all align with the cuneiform tablets that describe the 612 BCE date of destruction. So here we have some evidence of where there were cut marks on individuals. Some of these were clearly deadly, like a wound that was caused by something, a blunt-tipped object going through the abdomen of the individual here, and then other cut marks, even an arrowhead embedded in one of the tibia. We also have extensive evidence of perimortem fracturing on the bones, which some of these were probably more defensive in nature and others could be caused by the falling debris. Osteologically, based on the skeletal information, the adults were male. Based on bones, it's impossible often to understand whether the individuals were osteologically male or female. And so for the subadults, we did not know that. But among these male individuals, 4 out of 6 of them, these adults, they have evidence that aligns with being potentially their occupation was in the military. So things like markers on the hands, the elbow, the foot, and the other hand indicating that they were an archer. Another individual had extensive breaks on that had healed across the whole skeleton, including evidence of healed cut marks and things like that. So there were evidence among many of these individuals that they were possibly military individuals. The subadults obviously were not military individuals, indicating that they were likely civilians. So for this genetic data, the findings that we were able to extract from them, first of all, allows us to understand the genetic sex of these individuals. And so since we weren't able to see that osteologically, we can then understand, okay, we have adult males. Does that align with the genetic data? What about the subadults? Here we see that for the two that we extracted DNA, we can see that those were male, but the subadults included males and females. Okay, and also very interesting was that these were, none of these were genetically related. So you have individuals as young as 10 months, a 3.5-year-old, 7, 8-year-olds, 11, 12, this various age ranges, and none of them were genetically related. So the adult males, likely military people, the civilians who were the younger individuals included females and males, and all among them were unrelated individuals. Oh, okay, so it did go through. Okay, so those are the individuals that were sexed as genetically being male or female, and their ages. Okay, so now we're moving on to some of the genetic data that's going to take a second to unpack what we're seeing here. But this is a tool for visualization that's often used in genetics to be able to orient yourself to whether the ancestry of these individuals looks similar to sites that are either nearby or contemporaneously or geographically. And so we start with modern data that's underneath the gray. And then on top of those, because that data is better preserved, it lays a ground for the ancient individuals who are somewhat fragmentary in nature. But here we can see from the published examples, we selected the sites that were most relevant in terms of chronology and geography. And so any culture, any of the samples that were even close in the region near Nineveh. And we have one sample in particular that was very helpful in understanding the ancestry components that were present among the Nineveh individuals. Here we're plotting our new data. Our blue individuals are these individuals from Nineveh. And we can see that most of them fall into a main cluster, and that aligns really closely to that Nemric individual who's very close geographically. And then there's one individual who is more Levantine in nature. So overall, the individuals from Nineveh have a close regional shared ancestry. One individual is a bit of an outlier with a more Levantine ancestry component showing. That individual is number 8 here. That was a boy of 7 or 8 years old, and there were no other distinguishing features about this individual. He was part of this group, one of the subadults, but his genetic ancestry components do indicate some differences. Another individual here, number 4, this was a female of 11 to 12 years old. She has a distant relationship to somebody who she likely didn't even, not necessarily somebody that she knew was related to her, a distant relative who was buried in an Assyrian period tomb in the Levant. So this also shows a potential Levantine connection. But overall, we have a sense that this is a group of people whose ancestry components and had a lack of relationships outside of this area as well. So one of the questions that we're interested in answering that was specific to the Assyrian Empire is this practice of forced displacement, which some estimates put this practice at moving 4 million people across the empire. And as I showed you at the beginning, this was a large swath of land. And even it was focused on moving people very distantly across, and even as family units. There was also the practice from historical evidence of conscripting soldiers from other parts of the empire as well. And so the fact that these individuals show very strong local continuity from previous occupations of the region and also among each other, this clustering shows that there was likely not a huge amount of this forced displacement present among these individuals, although possibly with this single Levantine outlier that may have been part of his ancestry at some point in his family history. The uniparental markers, like these haplogroups, this mitochondrial DNA lineage, we do see a high level of diversity. And I'm showing you this just to give you a sense that although we had that regional clustering, we also see a lot of diversity among these lineages, showing us that this was probably a large population, which is in line with what you would expect from a large city. So Nineveh, the mitochondrial DNA does suggest that Nineveh was this large metropolis. This is similar to the Y lineages. There also is a high level of diversity, no repeats, just like the mitochondrial DNA. So when we look at this overall, we recognize that this is a very limited sample from a very specific archaeological context, but we do see shared affinities within the region and some continuity. We do see one outlier. Overall, we think that we see a metropolis with a diverse lineages playing into this population more broadly, and some gene flow from surrounding groups, but mostly a regional continuity. And then we also think of this moment in time with this exact individuals who are represented, showing a moment of chaos among these unrelated children and adults. So we recognize that this is a small sample, but we do think it helps us start to understand what are some of the processes that took place at the ground level for populations living in an empire that was known for grand cities and forced displacement. We see mostly regional continuity. Thank you very much.
We're going to hook up Professor Van Damelen next. Welcome to Professor Peter Van Damelen. Yay.
Yeah, can I get started?
Yep.
Oh, okay. And so you see my slides as well
Yep.
down there. Yeah, okay, perfect. Thank you very much. Well, good evening, everybody. And first of all, I'm sorry to not to be able to join you in person. The snow has kept me in Providence today. So I'm here virtually here to speak about a project that I've been a participant on. I've been contributing archaeologist on the project that has run over quite a few years, and that was published earlier, half a year ago or so, just before the summer. And you can see the details of that on screen. Punic people, the title was Punic people were genetically diverse with almost no Levantine ancestors. So I also want to thank Professor McCormick for the opportunity to speak here, and most of all, my thanks to the two of the main protagonists of this study, Harald Ringbauer of Leipzig, the Max Planck Institute, and David Reich, of whom you've seen earlier, for involving me for their in this study and for their collegiality and most of all fruitful collaboration. So I'll also just very briefly say that I'm an archaeologist, not a geneticist, but have collaborated in several of these projects for some time, and I think that has worked really well in this particular study. So first of all, if I manage to get my slides to advance. Something's happening. Yep. First of all, I want to begin with an archaeological comment, qualification, which is about the terms Phoenician and Punic. First of all, to say that Phoenician and Punic are not ethnic identities of any sort. They are labels, academic labels that archaeologists and historians have devised early in the 20th century and that have become well established as the conventional labels. So they're really academic, labels, as I said. And the main distinction between them is chronological. Phoenician refers to the earlier centuries of the first millennium BCE, and Punic to the later, to the middle and late centuries of the millennium, roughly with the sort of turning point being somewhere around in the mid-6th century, early mid-6th century or so. At the same time, there's also a regional component to it, which is that Phoenician really comprises the entire Mediterranean, not just the Western basin that you see here on the map, but also the Levant, Phoenicia itself. What you see on this map here is what I would call the Punic world and its constituent regions. So ranging from Andalusia in the west, sort of going up a little bit north in the Mediterranean, Sardinia, Ibiza, Sardinia, Sicily, Malta, and then, of course, there's Carthage itself. Yep. And it is these considerations, it would seem to underlie, or they're really critical for understanding the results of this study. Here you see the samples that were used in this study. That's also a really distinctive part of this study. 210 individuals who were sampled in 14 sites. You can see the majority of the sites in the West Mediterranean, really that part of the Punic world that I just showed, but also a good number of samples, 25 in all, in the Levant itself. In the east, and it's the samples from these sites that have given rise to this PCA plot, as you've seen in the previous presentation, and as you will see in the coming presentations. These are an integral part of these genetic studies, and what's really crucial for understanding the results of this study, I will illustrate with this. I'm going to make 3 key points on the basis of this plot. The first one that I'll try to visualize with this oval is that the elements in yellow that you see are the results from the West Mediterranean sites. The results in red above it, where it says Levant, are the results from the 2 Levantine sites. The other colors are other ancient samples that are there for reference. What's critical here, and that's my first point of interpretation, our first point of interpretation, namely that there is no overlap. There is, in fact, a strict separation between the Levantine samples that are the ones in the right-hand upper corner and the many yellow samples that are roughly contained within that oval stretching from the upper left to the lower right corner. As you can see, there's just no overlap between them. You can also see that those yellow samples, those within the oval, they are not clustered on a little lump, on a little pile together, but they're really stretched out, hence that I had to use an oval to sort of cover them. And they really stretch along from the upper part, where it says you see a few green dark green and blue dots, if you can make them out. But the upper part, it says Aegean, and those are samples from Sicily and the Aegean, the Greek world, that sort of are closest to that part. But then they also stretch all the way to the south, where it says Northwest Africa. And those are really samples coming from the Maghreb, from North Africa, and including the Canary Islands. And so they really sort of encompass a lot of variety that genetically was present within the Punic world of the West Mediterranean. The final point here is at the sort of far left-hand side. You see there's a few of those yellow elements that coincide with dark gray ones where it says Iberia. And those are samples from Iberia, present-day Spain, southern Spain, and that shows some connection and overlap with indigenous Iberian individuals. The bright green ones, as you can see, are totally separate. Those are from ancient prehistoric Sardinia, and you can see there's no overlap with that area there, despite that good number of the samples come from that island. To make sense of all this, it's important to turn to the archaeology once more. And here you see two Phoenician Punic burials. I'll begin with the one on the right, which is a Phoenician burial. And what's crucial there is that it is a cremation burial. The Phoenician world, the earlier centuries of the first millennium, preferably buried their dead treated their dead ritually through cremation, and then buried the remains with grave goods, as you can see here. The other photo next to it there is a complex of extensive chamber tombs, which is where people were buried, inhumed, bodies left intact, not cremated, in the Punic period. And that is really important to understand. These burials are found throughout the West Mediterranean world, but because of the strong prevalence of cremation in the Phoenician world, that is in those earlier centuries, we simply do not have any DNA evidence for that period, simply because cremation destroys the DNA within the bones, well, together with the bones themselves, of course. And so the first genetic evidence, the earliest genetic evidence that we can find in the West Mediterranean is of the Punic world, which is several centuries after the beginning of Phoenician colonization, of the first presence of Phoenicians after the foundation of Carthage. And that is really important, thinking of the title of the paper that I'm talking about. The research I'm presenting is it begins with Punic people did not have Levantine ancestry. That it is what we are saying is, well, what we are not saying is that the Phoenician people did not have any Levantine ancestry, but we are saying that the later Punic people had no Levantine ancestry. The reason for that is simple, is what you can see here in these two burial customs. We simply have no evidence for the population in genetic terms, at least for those earlier centuries. The only thing that we can see, as soon as we can trace those people back, those inhabitants, and can trace their DNA, we see that they did not have any Levantine DNA. That, of course, creates a conundrum, because Carthage, to begin with, and the entire Phoenician Punic world is well known to have Levantine ancestry. And again, the archaeology demonstrates that clearly. The Phoenician world, you can see here, Phoenician writing, Phoenician alphabet, characteristic red slit pottery, and the not so remarkable object on the other photo is a tanur, a little oven cooking stand. That's a very Middle Eastern type of cooking implement. This is just to say that culturally speaking, Phoenician heritage is very strong throughout the Phoenician and throughout the Punic periods in the West Mediterranean. But genetically speaking, by the time we can see, we can follow again. The Levantine origins appear to have disappeared, which, of course, the question, well, what does that mean? One likely answer for that is, well, that small numbers of people migrated originally and quickly mixed with increasing numbers of local and other people, and the Levantine signatures was, so to say, drowned out in the course of time. That is hard to prove because we don't have the evidence. But what it is important, there's a second point here that is equally important, if not more so, which is to say it demonstrates that genetic identity, genetic heritage, and material cultural heritage, as I've shown on this slide, are not necessarily intrinsically related to each other, and they can evolve and develop separately and go different ways, as is the case here. There's a really interesting parallel to be drawn here in the field of agriculture, sort of in the same area, and specifically arboriculture, the introduction of fruit and nut crops, in particular the grapevine. In a nutshell, grapevine and grape cultivation for producing wine was introduced in the West Mediterranean in close association with Phoenician sites. So pattern that has been especially well traced in Iberia, as you can see on this little map. The cultural roots of this introduction got really well established, as you can see on the photo, which is a double the lower basins of a wine pressing installation. You see a small reconstruction drawing of it there. This is much later. This is Punic from the 5th, 4th century, excavated in Sardinia. But it is this kind of installations go back to the Bronze Age in the Levant. And so it's clear, and they're very different from the kind of installations and implements that were used in the Greek world, for instance. So we can see this development. What makes this doubly interested is that it has been possible to identify in a couple of places the cultivar, the specific type of grape that was being cultivated, because in the whole of the West Mediterranean, grapes were indigenous and wild grapevines grew in many places. But genetic evidence, genetic analysis of a number of well-preserved grape hips has shown that there were also a new Oriental related associated cultivar was introduced, but it got mixed with local ones, in this case of simply through the process of grafting. And so we can really follow the evidence in this case. Well, it did not, through the grafting process, it did not get lost, but in the human DNA, we did lose track. Another element that's important to note here is that the evidence around the West Mediterranean is all based on Punic large chamber tombs of the type that I showed you that are associated with the major urban settlements, urban coastal and colonial settlements. There has been, over the last 10, 15 years, new evidence has been, especially in Sardinia, to some extent in Spain, southern Spain, has been found, which also shows that the Phoenician presence was not just limited to those colonial settlements. It is also to be found amidst major indigenous settlements further inland. And here you see one example of that from the site called Muraga Suraki, an excavation that I co-direct on the west coast of Sardinia. The rectangular building on the left of the aerial, you see, is a Phoenician house to all intents and purposes. Architecture, you can see the little detail in the inset photo. Architecture, mobile, portable material culture, all those other things that have been found there are sort of typically Phoenician. This house would not be out of place in Carthage. Yet it is found in an indigenous site and is actually very closely related to earlier settlement. You can see the heavy stone wall with the bigger stones in the inset photo, and you can also just about make it out in the aerial photo there. This Phoenician house is built right on top of an older indigenous style building. And it is especially this reconstruction that shows you what's going on. The big towered building is a so-called nuraga, a fortress typical of the Bronze and Iron Age indigenous Sardinia, nuragic Sardinia indeed, whereas the house in the foreground is the reconstruction of the Phoenician house that you just saw. So this is the situation in the 5th century BCE at this major indigenous site where Phoenician-style houses, people were living a Phoenician-style life, cooking Phoenician-style and eating from Phoenician plates, but all the while living literally in the shadow of this big indigenous fortress. We have no evidence of writing, of language from this place, but it shows you the sort of great complexity of heritage and cultural roots also in the past. And together with the genetic research that has been going on, we are finally getting sort of a grip on the complexities and subtleties, nuances of how people and peoples were living and co-living, cohabiting their lives in the past. Thank you very much.
So we move on now to a second presentation, which will be an introduction to a new project focusing on the same Punic world with newly collected individuals from a little different geological or geographical, I should say, in archaeological context from those which have been presented thus far in Professor van Domelen's work. And for that, we're going to have the privilege from hearing from a young archaeoscientist, Reed Morgan, now completing his PhD in the Department of History here at Harvard University.
All right. Thank you, Professor McCormick. It's a pleasure to briefly address you all and address some new data forthcoming. Unfortunately, I won't be showing results, but talking about a project that's expanding on the Ringbauer work that Peter just showed you to more intensively cover what's going on at the heart of the Carthaginian world in North Africa near the city of Carthage itself. So this work has been generously supported by the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean, and the sequencing is being carried out in Jena and Leipzig at the Max Planck facilities. The project is a collaboration between the National Heritage Institute of Tunisia, in particular with the director of Libyco-Punic Antiquities, Yamin Schair, and my advisors at the MPI, Johannes and Harold. And I just want to briefly point out, for us, a really important part of this project and ancient DNA work in these regions that really have been left off the map of ancient DNA has also been building up regional and local competence amongst archaeologists in Tunisia to be doing this kind of work. And so it was really a pleasure to be able to host Fatma with us in Leipzig to learn for a month the basic computational techniques, as well as to travel to Tunisia to run training on sampling techniques with young archaeologists working with the National Heritage Institute, as well as to be able to visit the sites that we're actually working on and see some of these necropolis. Peter already gave us a brilliant introduction to the history of Carthage and Phoenician colonization. So I'll skip over this, but briefly say, in terms of coverage of North Africa itself around Carthage, 2023 Moot's work showed the first 12 samples from Kerkouan, a Punic city that was destroyed in the Punic Wars and not reoccupied by the Romans. And then the Ringbauer data that you just heard about brought up the number of samples from North Africa to 34. But I'm very excited with the project we're presenting here to be more than tripling that coverage for Tunisia proper in the couple 100-kilometer radius around Carthage, and to be increasing the diversity of sites that are covered. So what I want to briefly just show you is how our sampling technique is trying to target places to get more of the diversity of geography, of social settings, and cultural groups within the Punic sphere with a variety of different burial typologies. So first off, we have 19 new samples for whom we had successful DNA recovery from the city of Carthage itself. As with the Carthage data from the Ringbauer paper, a lot of these are legacy samples. We've been hearing about the troubles of working with legacy data and the provenance there. So these are, while from the heart of the Carthaginian world, actually some that we have the poorest context for, and we're looking forward to seeing from the radiocarbon dating just what kind of story can emerge from those samples. Up the Majerda Valley, at the city of Jebba, we have 21 samples from an inland Numidian context. So whereas the rest of our samples all come from the kind of coastal fringe of Tunisia that was the most heavily influenced by Phoenician culture, we were interested in contrasting that with the genetic patterns that we see on these interior sites, and whether the same kind of demographic patterns reach in up the trade routes connecting into the interior of North Africa, or are a coastal phenomenon that we're seeing on the islands and the coast of Tunisia. We have one site from the north of Tunisia, El Mansoura, which sadly only had 2 individuals that yielded usable ancient DNA, but is an amazing site in terms of these narrow passage stairwells leading down to tombs that have amazing burial remains, showing those cultural links to the Phoenician world that were discussed previously. Souskaspa, the center of the second largest city in Tunisia today, yielded 2 samples from this period, and right down the way, Leptomenes, one of the important ports of the Carthaginian world, yielded 4 that come from a very distinct, different kind of chambered tomb, with these side chambers, larger front passage atria, that we also can see here on the top image at Thapsus, where we had 5 individuals. But Thapsus was interesting in that we were also able to sample a second necropolis that was exclusively infants buried in amphorae, to try and see if there's a demographic difference between the individual burials in the more expensive, opulent burial tombs by the coast, and the kind of infant necropolis on the interior. El Hakaima, we have 8 individuals that yielded data, and is interesting in that this cemetery has the highest percentage of multi-chambered tombs. So in terms of looking for the kind of kinship and ancestry profiles going on in this very distinct kind of burial, different from what we see in Jebba and the north, we're excited to see the results from here, in addition to one sample that yielded results from El Alia. So you can see a bit of the range that we're trying to cover of various burial typologies, social contexts, and geography, but I'm going to leave it there as a cliffhanger because we don't yet have publicly available results, so stay tuned.
Yay. Thank you very much, Reed. And now we're going to have the privilege of hearing from the genetic partner, and now the genetic archaeologist partner in our dynamic duo of Vicki Moses and Alison Barton. Alison is going to talk about some new ancient DNA findings from tombs in Cyrene in Roman Libya.
Thanks, Mike. And I should say I've been working very closely with Vicki, so even though she's not presenting this, she's been very involved. We've also been working with collaborators at the University of Chieta Pescara, and their collaborators in Libya, including some students who are local. And so I want to acknowledge them and all the work they've been contributing to this. They initiated this project and actually had reached out to our lab before Vicki and I even showed up or started this. So the site we're going to be talking about today, there's 2 that are in what is today Libya. The first is Cyrene, which is this coastal city that was a Greek colony. That's the story of how it was founded. In fact, the historical record says that there were migrants from Thera, which what is today Santorini, who established a city there, and as part of the larger Pentapolis region of the Greek state. We also have an additional necropolis that is called Ain Hafra that's nearby. It's a distinct archaeological site, but they're the same settlement. And then we have an interior site, El Jagbub, which is along the trade routes from Siwa Oasis in Egypt that run up to the Mediterranean coast. So we were hoping to compare the coastal and sort of the interior sites to see if we could pick up on any differences, both in terms of ancestry, population movements, and population size, things like this. I should also say that these are rescue excavations from urban encroachment and some environmental changes that have been going on. So we don't always have the exact context. Our dates are fairly rough because of that. We sent them out for radiocarbon dating, so we're hoping to hear back soon. But our best guess is that they're what we're broadly terming classical, somewhere in the Hellenistic or the later Roman periods in these regions. And to show you a little bit of what the site looks like, to zoom in, this is Cyrene on the coast and Jagbub on the interior, and this is our the necropolis at Cyrene, and you can see the tombs that are cut into the rock face of the cliff here. So another one of these principal component analyses that you've seen, this one is quite similar to what Reed and Peter have been showing you, and so what you see on this side is the Bronze Age Sardinia in Spain, Bronze Age Greece here, and the Bronze Age Levant on this side. And toward this end, we have individuals from Neolithic North Africa. And so this kind of gave us our background of the genetic diversity that would help us to visualize what we might see in Libya during these time periods. And so when we added our new individuals, I'll start with Jagbub, this interior oasis site. Most of these individuals represented by the triangles are shifted toward more toward the Levantine side of this. We call these a cline when there's kind of like a continuous line of individuals around here. They're very near to this recently published Egyptian Old Kingdom sample, which might make sense with what we see with the geography and it being along this trade route into Egypt. Then in Cyrene and Ain Hafra, we saw a lot more diversity. So our El Jagbub samples are all clustered quite close together, which says that maybe there's not a lot of genetic diversity. But in Cyrene, we saw individuals, some of whom were closer to the Punic individuals, although still shifted toward the Levant. We had individuals who are outliers closer to the Greek Bronze Age. Other individuals, two here that I'll actually talk about more later, so I just want to point them out, but they're shifted toward having a more North African ancestry. And then some others, such as these down here, that actually probably shouldn't be plotted on this PCA because their diversity lies outside the range of what was measured. And they appear to have mostly sub-Saharan African ancestry. And we're still investigating exactly where in that region. So just the overall impression was that there is a ton of diversity across this one site, this Greek city. Another way of visualizing this is with what we call an admixture analysis. And this is a way of trying to cluster components within a person. So getting multiple measurements of what their ancestries might be. And we don't give it a source. We don't say, try to match this one or try to match that one. We let the computer just cluster based on the math behind it. And what we see again for El Jagbub is that we see mostly this green, which is maximized in other Levantine populations here, as well as some blue, which is maximized in our other North African ancestry individuals. So these are the Neolithic Tunisian samples. And they also look quite similar to each other, which reaffirmed what we saw in the PCA, that we probably have a lot of homogeneity. And then again in Cyrene, the diversity really jumps out here, where you can see our two very sub-Saharan African individuals have this blue. There's quite a few that also have this orange component that's maximized in the Aegean and also Europe. With this level of discretion, we can't tell, but one of those two, but a high degree of diversity. And then in Einhofra, similar, mostly maximized by the Levantine, but with additional one individual who has some additional African ancestry. To follow up on this, we tested these identical by descent segments that Mike mentioned, and to briefly give a background on what they are. If you imagine this is a family tree, so this is two sets of grandparents, a granddad and a grandmom here. Everyone has a different genome represented by these different colors. In the next generation, they'll pass parts of their genomes on to their offspring, and they'll pass them on in turn to their own offspring. So if you took these two cousins, you might be able to pick up on some segments like this orange one, where they both inherited the same segment from the same grandparent. And that will tell you that they share an ancestor some number of generations back, and sort of how far back is dependent on how long that segment is. And you can see, for example, in the parents here, that this orange is much longer that was shared between them. So if we try to do that with our Libyan individuals, and we look around the globe and say, who might they share some of these segments with? What we see, some of it is not that surprising. So in Jagbub, we had some sharing with later medieval North Africans. You might expect that if they still have descendants hanging around in the region. In Einhofra, we saw some individuals sharing with individuals in the Italian Bronze Age. This doesn't mean that they necessarily came from Italy, but that they share a common descendant somewhere. And that common descendant could even be somewhere in the Aegean or Greece, for example, but there is something linking them. And then what was sort of most interesting was from these results was that one individual at Cyrene shares quite a few segments with individuals from Russia and Kazakhstan who were associated with Sarmatian culture. Again, doesn't mean that they were Sarmatian, or from the initial ancestry results, they probably weren't sort of 100% matching that genetic ancestry. But we also thought this was interesting because in this 2024 paper, the authors found that this individual who was buried in Roman Cambridgeshire was actually 100% Sarmatian ancestry and had isotopic data that actually linked them to growing up in the Caucasus region. So we are seeing mobility within the Roman Empire, at least, of Sarmatian individuals, so maybe a descendant here. We also see evidence for family connections within the sites. At Einhofra, we actually have a tomb system, and in one of the tombs, we see a father and a daughter and a second degree female relative of both. It's about second degree, so we're still trying to work out exactly how they're related. These portraits, too, are ancestral sculptures that were found nearby to these tombs, so they might have been associated ancestors with these individuals. At Cyrene, those two individuals that I pointed out that had additional North African ancestry, we actually see a brother and a sister, which has some interesting implications for mobility. If they're coming together, you might not think this is a mercenary. That could be that they were born there. We'll try to do isotopes to follow up on that, or they at least have a little more of an established family system at this site, despite looking like outliers, at least genetically. And in El Jagbub, we have a father, daughter, and a second degree male relative of that daughter. And another interesting finding at El Jagbub is that we have quite a few third degree relations, and we think overall this probably reflects the smaller community size in that more remote region. And so what we wanted to conclude on to kind of tie this together with what Reid and Peter also talked about is that there's this hypothesis that there's this east-west Mediterranean divide in terms of cultural contact and genetic contact, with the idea being that there's a divide kind of around here, so right between the sites we're looking at Cyrene, and the sites that they're looking at, here, for Carthage, for the Punic Empire. And the idea is that there is greater cultural contacts on this side, and then on this side of this divide across North Africa. Partially, there's a very large expanse of desert here, as well as like navigability reasons on the Mediterranean. And so we're interested in testing this, and I think this is a great place where the science of the human past and this collaboration can come together, because if you look back at this PCA with these Punic individuals and then our Libyans, you start to see that maybe there's some kind of division happening here. So hopefully our work can follow up and explore this further. So we have these distinct ancestries on the coast with some homogeneity versus maybe a more urban cosmopolitan setting that reflects the impacts of colonization. We see hints of this east-west divide, but we'll have to investigate when that arises and whether it's sharp or more of a gradient. And hopefully we've started to address some of these gaps in the data. So thanks to all our collaborators in the Right Lab and us.
Thank you, Allison. So to conclude our presentations, and as a prelude to the discussion that will follow, we're going to hear from both Allison and Vicky to offer some bigger result reflections and some really interesting reflections on some of the innovations, some new ways of thinking about finding historical archaeological connections across vast distances using genetic tools.
So I'll get started. As Mike said, what we were interested in doing was kind of being able to step out and look at the broader picture. So we've talked about some more specific sites or transects for specific empires, but we wanted to think about how can we start looking at mobility and movement and the effects of empire across these time spans and across larger geographic regions. So our question is, did these classical empires such as Rome impact the demography of local populations? We've talked about these identical by descent segments, and those can connect to individuals, across a quite broad time and space. And another way of thinking about this that Vicky will talk about is also just looking at ancestry and people who are outliers in their own sites. So we've taken a very broad view of the classical era just for these identical by descent connections because they can span thousands of years. But if we look at 4,000 years before present on this end, and about 1,500 to 1,000 years before present on the other end. This is sort of a map of all of the sampling that we've done with high enough coverage to start looking at identical by descent segments. And one thing you might notice is that it's very biased. We're missing a lot from the Near East. We're missing a lot from North Africa, which we're starting to fill in, but it's definitely not the same. And even some regions, such as France, for example, where you might expect more dense coverage just based on the amount of archaeology that's being done and things like that, we don't have this dense sampling. And so that can be a bit of a problem for IBD, but we're sort of trying to take a look either way. So if we look at these segments that are shared as identical by descent across this time span and location, if we look at segments that are at least 12 centimorgans in length, which we're pretty confident are true and that we know these two individuals are really connected at some point in time, we can see that there's just a wide degree of connections across all of these territories that were in contact during these time periods. In particular, a lot of this is due to the sampling density issue, but you can see quite a bit of connection between the Punic sites that we have as well as Iron Age and Bronze Age Italy, although how to control for that sampling is still something that I think a lot of us are working on. If you take that away, you can see a little clearer just for more examples. And this is an imperfect way, but I wanted to highlight some sort of intriguing stories that come out of this that we might have expected so far from the data, but hopefully we'll find new examples too. So one from North Africa, from the catacombs of Marcellinus and Peter, which is what this wall painting of Jonah and the whale is over here, because I put it in because Jonah was from Nineveh, and so to tie all the way back. So this individual was known to have North African ancestry in this paper looking at individuals from Rome. And we actually find that two individuals in some Roman Moroccan samples that we've taken a look at recently share more than 10 centimorgans of IBD with this individual. So sort of an intriguing line to follow up on. And another case study from the Punic colonies. This is a Punic ship that was found on Sicily. And this individual, who was also found at that site from Ringbauer et al. 2025, was a Levantine outlier. They found that in the paper. And we actually see quite a bit on these large 20 centimorgan segments of IBD sharing with two Iron Age Lebanese individuals. So these require broad sampling and multiple approaches. The IBD connections let us see some movements that might be difficult to detect from the archaeological record alone. But they're biased by sampling across all the regions that might be connected. The directionality can be difficult to determine. So as I was alluding to before, you could have a shared ancestor who might be Aegean, but then their IBD ancestors show up in Italy and they show up in the Punic regions. The timing is based on the size of the segments, but it isn't exact. And alternative approaches can be complementary. And I'll mention briefly too, this is all genetic. So this isn't like isotopic evidence that lets us see that someone has physically moved within their own lifespan too. But Vicky will talk about another approach.
So all of these various projects that we're sharing with you today start to encompass some of the work that we've done in this 2.5 years as Getty dual postdocs together. And one of our major goals, though, is to speak across disciplines and communicate these data to audiences that are outside of specialization. So have the archaeologists and the historians and the geneticists all able to communicate and get to interesting ideas together. And so one of the ways that we're doing that is to take the published data, and in this case, I'm speaking just about the Roman Empire, any individual who's been published, who's been associated with being culturally Roman, we're including here and mapping onto this space so that you can start to get a sense of where there are samples and dig into them further based on your own research interests. So this is part of a larger Science of the Human Past project called MAPS, Mapping Past Societies. And this is an interactive public tool that has many different layers to it, various archaeological, historical data, environmental data, and really anything that's spatial that you can dream up. And so here we're dreaming up the aspect of adding genetic data and making it as complete as possible, and correct as possible, but also making it something that's easy to consume regardless of what your background is. And so here we're starting just with how many individuals were published that are Roman, and what are their dates. And then moving on, you'll be able to, as we complete this work in progress, and we're very thankful, we've had a great undergraduate, Emily Ding, has worked on this, and I'll show you specifically her contribution in a moment. But you'll be able to hover over the sites and click on them and be able to see more contextual data, such as the site. You'll be able to look at the archaeological data, such as the grave goods, whether there's been isotope studies that have been done on that individual, what their orientation is or their proximity to other individuals, all these different things that can be laboriously added as various people have interest in digging into those topics, as well as just citations of where you can find more data if you have a particular interest area. And then finally, the genetic details. And so this is an example from the Balkan paper, the incredible work that recently come out from Alalde et al. And Emily Ding went through and identified what the ancestry summaries were for each individual to be able to just quickly summarize the best way that you could describe that individual's ancestry. And then this is a great tool to even say, if you want to look, as we get more data into this, looking at, okay, I just want to say all the North African outliers, all the people who were from the Levant more than other areas, and then being able to see them visually spread across space and see the chronology and be able to really play with this tool. And so this is definitely something that is a complex subject that we're trying to distill down into just a quick summary. So this is something that really takes specialized genetic knowledge to be able to do in a way that's true to the data. And as also part of our project, which not only is hoping to be able to increase communication across many fields, we also wanted to increase the sampling coverage and make sure that various cultures and time periods are represented. And so we've been lucky to work with museum collections, work with legacy data, some of which was been ignored and buried in museum collections for a long time. We're really happy to have those collaborations in place. Working with things that have been processed years before and have just are now getting analyzed, or things from active excavations. We're really thankful for the people who have hooked us up with some collaborators or samples. And so now we're able to add, for even just the Roman period and other periods in the classical antiquity, Moroccan samples, Tunisia, Libya, Israel, Jordan, Turkey, and Iran are part of the areas that we're able to add into this. And so we're very thankful for those collaborators. And this is all really thanks to the Getty funding that's supported us over these last couple of years. And also just a range of scholars who've done things just from one conversation to extensive help or connecting us to additional knowledge. And so we're really grateful for all the people who've been instrumental to this. A special thanks to those who have guided us through this. Professor McCormick and Professor Reich have been, this has been a very fruitful conversation. So with that, I'll turn it over to you. Thank you.
Thank you both. Thank you both. Thank you, Peter Van Damelen, for joining us, and thank you all for coming out in the climatic, challenging time in which we are. And now it's time to turn the floor over to you for your questions and observations, the ideas that you might have to share with our speakers. So let's have at it. We'll ask you just to come up or we'll hand you a microphone and ask you to identify yourself and pose your question. All questions are allowed. We might not have answers, but we want to hear the questions.
Hi. First of all, thank you to everyone who presented today. This was wonderful. I'm a sophomore at the college studying ancient history. So my name is Henry, and I'm not super familiar with the DNA aspect of this. So this has been wonderful getting to learn and seeing the principal analysis. I guess the main question that came to mind on a very large scale is I can sort of picture the, for lack of a better word, traditional classicists and historians writing a bunch of defensive articles after this saying, what are we going to do with all this genetic information? Genes don't make a person. This doesn't tell us about culture. And we need to go back and problematize all of this. And so I guess, how is this work going? Do you envision it sort of aiding or questioning traditional narratives in classics and history? And how do you imagine sort of people like me who don't know anything about DNA using this work in the future to guide their own research? Thank you. Sorry, I hope that wasn't...
No, that's a wonderful question. That's a wonderful question, Henry. Team, you want to take a crack?
Sure. Yes, I think that's a really big topic because it is a potentially fraught line of evidence that if you don't think about it and what you're implicating by talking about populations or equating your genetics to your identity without questioning it further, I think you have to just take it as another line of evidence that deserves just as much rigorous skepticism as ceramics or architecture or an animal bone that's nearby. So just like any other line of evidence in archaeology, you have to be very careful about how you use it. And I do think that, especially given the different fields that are coming together and the different scales that we usually work at. So historians or archaeologists, we tend to work on very site-specific, individual-specific levels, and then genetics does tend to be broader. It is hard to get the data and the perspectives to line up, but it's something that I think we're all pretty aware of. And with things like this, where we're trying to have interdisciplinary work, we're able to address them a little more. But it's very difficult process.
Yeah, and I think like any type of archaeological or historical data, I mean, we know historical records are biased, so are the genetics in like their own certain ways, right? And so I think it's just important to know the limits of what you can say. And sometimes the results go to upend ideas about people. This idea of genes not being related to culture necessarily. Sometimes you can show that it isn't. Like in, the Punic example or Cyrene, you have people being buried in classical tombs, but obviously they're very diverse genetically. So obviously the culture is not tied to one ancestry, or also upending ideas about kinship, for example. Vicky had sort of an example of this with Halsey Gate. She also worked on a paper on Pompeii, where people thought children must be related to the adults who they were found with, and that actually wasn't the case. And so I think you can make a lot of different conclusions from it too. Yeah.
One thing I would add is that, or emphasize, is that this is all independent information to the archaeological information or historical information that we already have. And it gives us the privilege to visit archaeological sites and features and contexts that have been carefully excavated and published, and to suddenly have a completely new piece of information. Like, for example, you remember in the 2023 Olalde paper from the Danube, there's a young man, 18 to 20, died in the Roman provincial capital of Imanacium on the Danube around 100, well, between 100 and 200, buried like a normal Roman with a nice little oil lamp with a spread eagle depiction on it, and turns out his ancestry is entirely Somalian. This was quite unexpected. And so it's a completely new piece of evidence in each case that obliges us to go back and to rethink, and sometimes gives completely unexpected new meaning to finds that look to be quite ordinary at first blush. So it's an excellent question, and there's a lot to be done ahead. We have to do it carefully and scrupulously, as we do with our archaeology, as we do with our philology, step by step. Professor Ur.
This is very exciting to see all of these new results, especially with the genomic or ancestral diversity that's emerging across regions. I'm really interested to see how this diversity is going to be playing out within sites. And this, I think, is really important for thinking about empires, because empires promote a kind of cosmopolitanism that makes cities diverse. And, you know, Alison, this is something that you started to kind of get into in your Libyan case studies. And so this, in thinking this way, I'm thinking of Vicky's case study from the Halsey Gate in Nineveh, where there's a remarkable, I think, uniformity of ancestry amongst most of the individuals that were found there. And I think it might be tempting to say, well, this is Nineveh. But I would actually encourage you to think of this is the neighborhood around the Halsey Gate. And it raises all kinds of questions to me about what does the rest of the city look like? Now, obviously, we don't know yet. But here's a case where if excavations could return, targeted excavations perhaps at the other gate might reveal might reveal similarly uniform kind of genomes, but entirely different from the Halsey Gate, which might tell us something about the ancestral diversity within Nineveh. Is that something that we can do yet, either in settlements or in kind of mortuary sites? Is that something that's in the future?
Yeah, that's a great question. I'd be interested, like, I know Reed's in the audience, but if you have anything to say too, but I think part of the way we have to answer that is by having samples that we know the like in situ context for. And I think there are some projects, I'm thinking of later time periods where we have cemeteries where we have, you know, patches and corners, and maybe we can start to see some of that, like you've seen in Harvard and Cambridge. I think what we've been kind of constrained by is having a lot of, like in Libya, having a lot of the remains disturbed. And that's true in, I work on some projects in the Americas too, and there's a lot of secondary disturbance of the material. But I think as we are able to sort of have these larger and larger projects, we are going to be able to get into that direction, which would be super interesting.
Yeah, I think it's
Your wish is my command.
individuals. Can we even say that they're from that neighborhood? That's even a question mark. And so you do the best you can, and then we're getting new data all the time. And what we can say now, I mean, we're rewriting things all the time. And so you just have to eat your words a little bit sometimes and say the best within your limits, and then things might change.
If I can just maybe briefly speak to that, I do think we're at an exciting moment where ADNA is got big enough and enough data that it is now actually shaping fieldwork agendas. So the Max Planck Institute has started running an excavation in Morocco that's targeting filling in a gap. And I think we're increasingly going to see moving from geneticists with research questions together with archaeologists looking at the available data to excavations being started to fill in gaps.
Thanks.
All right. I have a question, actually. Sorry. My name is Ian Wilson. I'm a PhD student in classical archaeology. And my question actually has been set up really excellently by our previous speakers about gaps and cosmopolitanism. I'm wondering about population centers in the ancient world. Particularly, your map highlighted, notable gaps and like future agendas where you want to build up this data. But I'm thinking particularly about like Egypt, and I know Egypt has its own problems. But what concerns do you have about your data sets and your models when you can't factor in a city like Alexandria or like one of these other really major centers of population and also of like cultural fusion? Is that a question that matters?
Oh, no, that's a great question. I think it goes back to this knowing the limits of what you can say. And I think for Libya, the one sort of very old Egyptian genome we have actually came out while we were working on the samples because, as you alluded to, for political reasons, it's difficult to sequence individuals from Egypt. But I think, yeah, you just have to account for that. So we sort of figure that there might be this connection, obviously, especially for this site that's along this trade route. And so then I think you just have to leave that possibility open when you're interpreting it. Yeah, that's a great point.
And I think that's part of the importance of having these conversations where if you should be aware that a city like that exists that could play into that. So the conversations between the archaeologists and historians and geneticists are critical. I have a question for Reed. Do you have a sense of why El Mansoura had such a poor preservation rate? Was there something about the deposit?
Nothing obvious to me. Yeah, preservation, as we know, is extremely stochastic, and it's a coastal site, like Carthage, like many of these. It could be something about the structure of the tombs that they're in. These are contexts that had air exposure and are in small chambers, but a lot of the chamber tombs from the south yielded better. So yeah,
who knows?
No good hypotheses there yet.
I'll make a comment on that. I mean, I'm not a wet lab person, but it really is a mystery.
Archaeogeneticist Nick Patterson.
Yeah, it really is a mystery. For example, there's this magic cave in Siberia, Denisova Cave. There's a magic cave in Siberia, Denisova Cave, and every fossil we get there produces immense amounts of DNA. I mean, for example, the very first fossil, the famous Denisovan, was a small joint of a child's finger bone, right? And it got immense. The coverage was pretty much as good as you would get from a modern sample in a high-quality lab. About 120,000 years old. And recently they excavated another fossil there, which is more than 200,000 years old. It's another Denisovan. The DNA is absolutely superb. And then on the other hand, you take the Indian subcontinent, we can get almost nothing. And nobody knows why.
Yeah, I guess just to jump in about why this region sort of has undersampling. For a long time, I think we thought we knew what made for good DNA preservation. And as the technologies increased, I think we found we don't totally know. So these regions are hot and arid, and we thought that was bad, but sometimes we were getting things. We used to think tropic regions didn't preserve anything, but that's also been proved not true in like the Central Americas too. So I think we're still kind of on the edge of understanding why we get good or bad preservation.
We have a question from the chat.
Yeah. Cool. I'm speaking for Edie Park, who is tuning in remotely and wants to ask, can genetic evidence shed light more on the stories of individuals or on patterns of migration and colonization within ancient empires more broadly?
I guess I would say both, probably. I think we need some of this broad sampling in order to figure out what the background is and what things might look like. But there's some really interesting individual stories I mean, Mike, you described this story about the East African individual who was buried on the Danube. Of course, we need some reference individuals to know that that individual is from East Africa. There's really interesting stories. Megan Michael, who's here, presented on an individual who had malaria at high altitudes in Nepal a year or two ago, which is like obviously an individual story, but really interesting. And then I think we saw examples of with this Punic paper of how we can trace empires too. So I think it's a really valuable tool for archaeology.
Both.
Yeah.
Individual and population. Individual mobility, for example,
yeah.
And population wildlife.
Yeah.
Evening is advanced. And
you are a wonderful audience. It's just amazing you're here through all the sleet and snow that we've been through in the last 24 hours. I think that you have well earned to continue the conversation over a nice glass in the reception. We wish those of you who are here joining us online could join us for that. But let's, in the meantime, thank our speakers one more time for all they shared with us.